Hello, everyone. Thank you for taking the time to join our webinar. So this is the first one of our Researchers at Work webinar series, Essential Guide to Becoming a CRISPR Cas9 Expert. Today's topic is CRISPR, the future of biotech. For those of you who are new to our webinars, they are designed to provide constant and systematic training that will keep you well informed on our products and services. After the webinar, we'll be sharing a copy of the PowerPoint slides as well as the recorded webinar itself. I would like to point out that if you are signed into a Google account, there is a chat box to the right of the screen where you can ask any questions you may have. At the very end of the webinar, we'll be answering your questions as many as we can. Please note that if you did not register for the webinar earlier, there is a registration link in the video description. This will ensure that you get a copy of the slides and answers to the questions posted in the chat if we are unable to answer them all within the webinar. Before we begin, I will introduce the speakers first. Joining me today is my colleague, Mr. Bashi. He's working for ABM as a product specialist and has multiple years of R&D experience in molecular biology. Alongside him, you have me, Nancy Yao. We're both involved in product development and commercialization of CRISPR Cas9 kit at ABM. Our goal is to provide you with the support that ABM can offer. Before we begin with the core content of the webinar, I would like to take a moment to talk about our company. Applied Biological Materials, ABM, is a leading biotech company headquartered in Vancouver, Canada. Founded in 2004, the company has been catalyzing scientific discoveries in life sciences for the last 15 years. Our complete portfolio of products and services have made us an invaluable and reliable partners to universities and research institutions around the world. As we grow and expand every year, we open a second branch in Zhenjiang, China in 2013, and very soon in the future, we're going to open a third branch in Bellingham in the US. With a team of passionate and trained scientists, ABM is dedicated to empowering researchers with the latest innovation for all their research needs. Before we discuss how CRISPR can be used as an experimental tool, it's useful to discuss its origin and power in terms of human advancement. 2012 was a year when CRISPR-Cas9 system was first used as a gene editing tool. Jennifer Doudna, Emmanuel Charpentier, and their research groups published their successes in precisely cutting double-stranded DNA in bacteria. However, it wasn't until the following year that CRISPR-Cas9 was successfully used as genome editing in human cell cultures. This work was done by Feng Zhang and George Church from the Broad Institute of MIT and Harvard. Recent studies have shown the advantage of CRISPR with regards to food production. One example focused on increasing the yield and size of tomato that are high in antioxidants, while a separate study was able to provide rice that produced viable seeds, allowing farmers to avoid buying new seeds every planting season. This demonstrates just how powerful CRISPR is, as we can eliminate and reduce hunger around the world by increasing the yield of our crops and making it easier to produce new crops. Since its inception, the genome editing market boomed within the scientific community. The number of publications related to CRISPR technology has been increasing dramatically for the last decade. It doesn't seem like it will stop anytime soon. To meet the falling demand of CRISPR, ABM has developed a comprehensive portfolio of CRISPR products, from Cas9 proteins to CRISPR knockout cell lines, all designed to help you with your experiments. With that being done, I'll pass it over to Bashi, who will continue the webinar with an introduction to CRISPR.
Thank you for that fantastic introduction, Nancy, and thank you to everyone for joining us this morning. As Nancy mentioned, I'll be starting the core content of the webinar and would like to begin with an outline of what we will be covering today. We'll start with some introduction to CRISPR itself, then we'll go over the entire workflow of a CRISPR experiment and how ABM can help each step of the way. Please know that we carry all the tools mentioned today, so please talk to us if you need to identify a catalog number or if you'd like to get pricing for an item. There are two main components needed to perform CRISPR, Cas9 and sgRNA. When I talk about CRISPR, there's an analogy I like to use. Imagine that Cas9 is a knife that can cut DNA. The sgRNA is a hand that guides the knife to where it needs to go. Together, they come together to cut genomic DNA. But what does that cutting look like? Here you see a diagram of the sgRNA guiding the Cas9 protein to its corresponding target sequence. You can't just target anywhere in the genome, though. The target site must occur next to a particular sequence called the PAM sequence. The PAM sequence is different depending on what type of Cas9 you're using. So, now you know that CRISPR cuts DNA. But how does this cause a gene knockout or knock-in? On the left, we can see what happens if we only have Cas9 and sgRNA without a repair template. In this case, the cell will repair the cut DNA imperfectly, causing the insertion or deletion of some nucleotides. This can lead to a frame shift of the translational reading frame, leading to the formation of a premature stop codon. The protein created will be truncated and non-functional, so the gene is knocked out. However, if we provide a repair template, a knock-in can occur instead of a knockout. The cell will use the sequence you provide as part of the repair template to repair the cleaved DNA, leading to the knock-in of that sequence. Now, let's go through the overall workflow of the CRISPR experiment. The first step is to select a Cas nuclease to use. There are many to choose from, each with their own advantages and disadvantages. Step 2 is to design and clone your sgRNAs. SGRNA design can be tricky, so I'll talk you through some of the factors you'll need to consider. The next step is to select your method of delivery of your CRISPR components. There are many to choose from, so I'll discuss the pros and cons of using each system. Once all the design work is completed, the next step is to actually perform the experiment. Finally, the last step is to verify that your cells were edited correctly. I'll discuss 3 methods for CRISPR validation and when you might want to use each. By the end of this presentation, I hope to show you that ABM is there for you every step of the way. We provide the products and support necessary to carry out a CRISPR experiment from start to finish. Now, while I showed you the workflow of a CRISPR experiment, a fair question to ask is, how do I choose the right tools for the job? This can be a large task to handle, but as always, ABM is here to help make your life easier. If you aren't sure where to begin your project, you can always start with our easy-to-use CRISPR experimental design tool. Don't worry about writing down the links at the bottom of the screen. All links in the presentation can be found in the video description. This tool streamlines your experiment by asking you to answer a few multiple-choice questions regarding the type of experiment you plan to run, the model organism and system you'd like to use, and your preferred integration method. As you can see here, I've selected a gene knockout in either a human, mouse, or rat using an immortalized cell line with stable integration of Cas9 and sgRNA into the genome. From this, we recommend the tools you'll need to run your experiment, from your source of Cas9 all the way to the recommended experimental validations. This can be very helpful in that it will save you from some of the more common errors in running a CRISPR experiment, such as choosing the wrong delivery system. For example, if you're looking for integration, then you would need to use a lentivirus. On the other hand, if you're looking for transient expression, you would want to choose an adenovirus. By taking advantage of ABM's CRISPR knowledge base, you can arm yourself against these errors. With that being said, though, let's start with step 1 of our workflow, selecting a Cas nuclease. You'll see shortly that ABM carries a wide variety of Cas nucleases for every application, such as SP Cas9 nickase and nuclease, which are capable of creating 1 or 2 cuts respectively and are derived from Streptococcus pyogenes. Cas9, which is a smaller version of the Cas9 derived from Staphylococcus aureus. CPF1, which contains T-rich domains and is derived from Francisella novicida. And finally, Cas9, which is a null mutant of Cas9 that can be used as both a negative control and as a tool for reversible knockdown of gene expression via binding. As we mentioned, there are many types of Cas9s to work with. This chart can help you choose which Cas nuclease is right for you. Cas9 is commonly used and affordable, but has a high chance of off-target cutting and is quite large, so it limits your choice for delivery. Cas9 nickase is a mutated version of the Cas9, which is used to limit off-target cutting, but requires 2 sgRNAs and low efficiency. Cas9, seen here, is a miniature version of the Cas9, which allows it to be delivered from small viruses such as AAVs. However, Cas9 is less commonly used and has fewer potential target sites due to its long PAM sequence. CPF1 is a bit different from the other Cas nucleases. When it cuts, it creates sticky ends on the DNA, which can be used for specialized applications such as cloning. It also has a T-rich PAM sequence, which allows it to be used in AT-rich genes, unlike other Cas nucleases. However, because that PAM sequence is so long, it is more difficult to find targets. ABM also offers dead Cas nucleases, or Cas9 enzymes. These are called dead enzymes because they can't cut the DNA, they can only bind to it. However, we can attach domains like the SAM or the CREB domain, in order to activate or repress genes. With that done, now we can move on to step 2, in which you'll need to design and clone sgRNAs for their use with Cas nuclease. ABM offers pre-designed sgRNAs for any gene and any application. SGRNA design can be a difficult prospect. The target must be next to a PAM sequence, and each Cas nuclease has a different PAM sequence. You can see that the PAM sequences can be quite different depending on the Cas nuclease that you're using, shown here on the right side of the screen. As well, the target site will also depend on what the purpose of the CRISPR experiment is. When designing an sgRNA for gene knockout, you should target an early exon of all transcript variants of the gene. For knock-in, the target site should be within 30 nucleotides of the desired knock-in site. Activation and repression require the sgRNA to target near the transcription start site, while base editing requires the target sequence to contain the target nucleotide approximately 14 nucleotides upstream of the PAM sequence. There are other factors to consider as well, including the sgRNA's GC content, length, scaffold sequence, and which promoter to express the sgRNA from. Poor sgRNA design can lead to off-target cleavage or no cleavage at all, causing your experiment to fail. For this reason, we make it easy for you when it comes to design and cloning. We offer a full library of sgRNAs for any human, mouse, or rat gene that are designed to target the 5' end of early exons to prevent the formation of partial gene products that may still be functional. Specialized software is used to design the sgRNAs with the highest predicted cleavage efficiency. However, in practice, there are many factors such as cell type and availability of the target genomic region that may reduce cleavage efficiency. To counter this, we recommend that clients purchase a set of 3 sgRNAs that will each target a different region of the gene, maximizing the chance of a successful knockout. We can also design custom sgRNAs for any gene or purpose you may have. In the past, we've designed sgRNAs for use in dog, fungi, and bacteria. As well, we can design sgRNAs not only for gene knockout, but for activation, repression, or other specialized functions. Also keep in mind that all of our sgRNA products include us taking care of the design work for free. Now that we have our components selected, we need to decide how to best deliver them into our cell. You already know that Cas9 and sgRNA can cut genomic DNA, but genomic DNA isn't just floating around, it's within the nucleus. Therefore, we need a way to transport those components into the cell. I'll now walk us through some of the ways we can deliver your CRISPR components into the cell. Vector transfection is an inexpensive and easy way to express your components, but this can be an inefficient process. Or you can package your vector into a virus. Different viruses have different strengths. Lentivirus is a commonly used choice, as it can cause the genes to be integrated into the genome, leading to long-term expression of that gene. Adenovirus causes short-term expression and has high infection efficiency, but can elicit a strong immune response in vivo. AAVs also lead to short-term expression without the immune response in vivo. However, it's a small virus, so it can only fit the SA-Cas9, not the SP-Cas9. If you're having trouble deciding what type of virus to use, feel free to check out our vector selection tool linked at the bottom of this slide. RNP transfection is another method you can use where nothing is expressed by the cell itself. Instead, you'll deliver the Cas9 and sgRNA directly as an RNP into the cell. Because they only remain in the cell for a short time, there will be fewer off-target cuts made when this method is used. However, it can be difficult to optimize for some types of cells. Finally, for Cas9 expression, you can also use a cell line that stably expresses it. This is a bit pricier, but the cell line can be reused for multiple experiments. If you would like to use a vector or virus format, you can now choose between a one-component or two-component system. The all-in-one system causes expression of sgRNA and Cas9 from the same vector, which simplifies the workflow and is less expensive than purchasing two separate vectors. However, it means that the vector can't be reused between different experiments. It should also be noted that there is a puromycin resistance gene included in the vector. This acts as a selection marker and can be used for confirmation of a successful CRISPR experiment. Two-component systems split the expression of sgRNA and Cas9 between two vectors, or between a vector and a Cas9 expressing cell line. This method allows you to experiment with different sgRNAs more easily, and the Cas9 component can be reused between experiments. No matter what delivery method or format you choose, ABM has a product that will fit. For more details, you can always send us an email or ask questions in the Q&A, and we'll do our best to provide more answers. Now that we've completed the experimental design and have our components ready, step 4 is to actually perform the experiment. As I mentioned, performing a CRISPR experiment involves delivering Cas9 and sgRNA into the cell you wish to edit. If you don't have an appropriate cell line, though, don't worry. ABM offers thousands of cell lines, and we can help you find what you need. Now, in terms of running the experiment, we can streamline the process down to 3 components: transfection and transduction, selection, and screening. In order to make your experiment as simple as possible, we're happy to provide you with a free copy of our CRISPR Knockout Manual, which provides a detailed protocol for conducting your experiments, as you can see on the right side of the screen. As you can see, this manual was designed to aid researchers who may be new to the field of CRISPR by walking you through each stage of the experiment and providing step-by-step protocols for you to use while you're at the bench. Now we've reached the last step of our workflow. We've done the CRISPR experiment, but now we need to see if it actually worked. I'll walk you through 3 methods of CRISPR validation. To answer the question, was the cell line edited? ABM offers 3 different tools and services: the Mismatched Cleavage detection assay, Sanger sequencing, and a next-generation sequencing service. The mismatched cleavage detection assay is a fast and inexpensive way to screen many clones at a time. However, this surveyor assay has low sensitivity and doesn't give any information about the sequence of the edited gene. I recommend this assay as a preliminary screening method if you have many clones to screen or a mixed cell population. Sanger sequencing is an inexpensive method to get sequence information about the edits of a monoclonal cell line. However, using Sanger sequencing on a mixed population of edited cells will lead to an unreadable trace, as seen here. This means that the Sanger sequencing of a mixed population can only give you a yes or no answer as to whether editing occurred, but doesn't give sequence information. Now, why does sequence information matter? Because not all types of edits will lead to a gene knockout. For a gene to be knocked out, it must have a frame mutation on both alleles. I recommend Sanger sequencing as an inexpensive way to confirm that a monoclonal cell line has had the gene fully knocked out. Next-generation sequencing is expensive, but it has many advantages over other validation methods. It provides quantitative sequence information about both monoclonal and mixed cell populations and is highly sensitive. It's possible to pool and screen many monoclones together at once for a high-throughput approach. As well, by using whole genome sequencing, you can check for any off-target editing which may have occurred. Next-gen sequencing is a great choice to validate a mixed population of cells or many monoclones at once. It's also a robust way to check for off-target editing, which can confound experimental results, especially in vivo. Here we have a nice summary table of the three techniques showing the benefit of each. We can see that in terms of sensitivity, NGS reigns supreme, but at the same time, the cost increase in terms of sensitivity is substantial, with mismatch costing approximately $6 per reaction, Sanger sequencing costing $150 per reaction, and NGS being about $600 per reaction. As such, I would recommend that you begin your validation with the mismatched cleavage detection kit and proceed on to either Sanger sequencing or NGS, depending on your needs and the success of your mismatched cleavage. And with that, we've gone through the workflow for an entire CRISPR experiment. I hope I've demonstrated how ABM can provide everything necessary for a successful CRISPR experiment, all the way from experimental design to validation. Now, one question that may come to mind during this is the time cost. From what we've seen, a typical CRISPR experiment consists of multiple steps, which can take approximately 2 to 3 months from the planning stage to knockout validation. With projects like these, it's important that we make sure to understand how to best optimize the experimental process and minimize the number of potential roadblocks we can experience. And don't just take my word for it. We have many publications and testimonials from scientists who have succeeded in their CRISPR experiments with ABM's help. The data shown on the right side of the screen is from an experiment where we completed the work using our services. We also offer a knockout cell line library where you can choose to have any human gene knocked out from any of our popular 8 cell lines, seen here. In addition, we offer Cas9 expressing cell lines to help jumpstart your experiments. Or if you have a particular cell line in mind, we also offer custom CRISPR cell line services where we'll do the knockout in your cell line. We are very confident in our CRISPR expertise, so recently we've also started offering a guaranteed knockout service where we promise we'll knock out your gene, and if we fail, you'll get your money back. With that, I'd like to pass things back to Nancy and let her wrap up the webinar.
All right, thank you, Ashi, for the wonderful talk, and I hope you all found it as helpful as I did. As a thank you for joining us during the webinar, we want to help you by offering you a promo code of 10% off on your CRISPR products, which will be included in our follow-up email and also in the video description. Before we go into Q&A session, there are some resources that ABM offers to help you with your CRISPR research. Firstly, make sure to check our website at info.abmgo.com/CRISPR, where all of our CRISPR resources are collected. These include our project design tool, the knowledge base articles, as well as knockout handbooks and case studies. All tools are meant to make CRISPR easy for you. In addition to these resources, we also offer free CRISPR crash course that will help you walk through all the theory needed to run your own CRISPR experiments. The course is broken into 4 sections that mirror the steps that were discussed today, including experimental design, delivery, and validation. If you would like to register for the course, please follow the link at the bottom of the slide. Not only do we have a diverse selection of educational CRISPR material, we also have an incredible technical support team who can guide you through your experiments, as well as a dedicated customer service team to ensure that you receive your items in a timely manner. If you have any questions about our product and services, you can always reach out to us by phone, email, or chat. Feel free to check our FAQ on the website as well. All right, so thank you all for joining us during this session, and I hope it was helpful for you all. With that being done, we'll begin answering your questions. It looks like we've got quite a lot of the questions. I guess let's start with this one. It's from Ian O. And the question is, how is CRISPR better than siRNA? Bashi, do you have an answer for that one?
Yeah, that's a really good question. So the main difference between siRNA and CRISPR is that siRNA works by degrading the mRNA, while CRISPR works by changing the DNA. Some mRNAs will escape the siRNA and can get translated into proteins. That's why when we run a CRISPR experiment, it's called a gene knockout. But when we do an siRNA experiment, it's called gene knockdown. So with siRNA, you usually get something like about 80% gene knockdown of the gene. But when you run a CRISPR experiment, if everything has worked properly, you should get 100% knockout of the gene. And that's why more people are tending to use CRISPR now than siRNA. Hope that helps, Ian.
Okay, thank you, Bashi. Let's see what's the next question. Okay. The second question is from Tracy Miller. She's asking how CRISPR can be used on long non-coding RNAs.
Yeah, Tracy, it definitely can be. I know we didn't mention it earlier, but we do have a specific service for that. If you're interested in doing it yourself, we also can help with the SGRNA design. But the hard part of doing sequence deletions is that the efficiency is much lower than with regular knockouts. For sgRNA design, you want to target the start and end of the long non-coding RNA with 2 sgRNAs so that the DNA gets removed, because a normal frame shift isn't going to be able to do that. Because of the low efficiency, you may need to optimize your targets. I would probably recommend trying to use multiple sgRNAs, either 2 sets of 3 sgRNAs or a multiplex vector with 4 sgRNAs, so that all of them get expressed at once. And then, depending on how long the deletion is, you could probably screen for it using PCR to look for a smaller amplicon. Hope that helps, Tracy.
Okay, the third question from Jansky. So the question is about designing sgRNAs. What kind of controls can we use for?
Jansky. So anybody who's running a CRISPR experiment should always be thinking about the types of controls they would need to use. So we give you two options. The first is our scramble control, which comes in both our all-in-one and our two-vector system. Because the scramble control ensures that the sgRNA can't find the target sequence, you can ensure that all the other components of your experiment are working properly. The other control option is to use our Cas9, which I mentioned earlier. It can't cut the DNA, but it can bind. So that way you can ensure that everything is working you can make sure that everything is working and still binding in the right area. So those would be the two controls that we would recommend. Thanks for that great question, Jensky.
Okay, let's see what will be the next. I think we are running low on time. Probably we'll just take one more question, but you are still welcome to leave all of your questions in the chat box and we'll answer them in the follow-up email. Okay, I think we got our last question today is from Alex N. So what is the difference in genome editing between primary and stem cells?
That's a really good question, Alex. So the key difference between the two of them is the method of delivery. RNPs are known to be better in delivering genetic material into stem cells than they are into primary cells. I hope that's helpful.
Okay, I think that will be the end of the webinar. And thank you all again for joining us today. We'll send out an email with a copy of the presentation slides and the recorded webinar, as well as the Q&A. And also, I would like to mention again that this is the first one of our Researchers at Work webinar series, and there will be more coming up along the year. So please subscribe us on our YouTube channel and follow us on our social media to get the most up-to-date information. So that's it, and we'll see you next time.